All right, good afternoon, everybody. Welcome back. It's nice to see everybody again, or at least see all your names again. Hopefully, everybody's been studying very hard since we last met, and I hope you all took a few hours to enjoy the snow today. I'm actually Canadian, so I am just loving this today. I'm completely in my element. Our whole family here is just having a blast with the snow this morning. So we have an exam coming up. I'm sure you're all aware of that next Tuesday. So I thought before we do start the lectures today, and I'm going to be lecturing today. I thought we just spend a few minutes talking about the exam a little bit, and I'll respond to any of your questions that you might have about it. So I talked about the exams right back at the beginning of the course. We do have three for the course, equally weighted. This exam is going to be 50 questions. All right, 50 questions, multiple choice, and they're evenly distributed through the sessions. So we've had 5 sessions. So you can expect, 10 questions coming from each session. So Dr. Humayun lectured for, a total, his microbiome was split between 2 sessions, but, because there was 2 parts to it, you can expect, for example, 10 questions from him on the microbiome material and, 5 questions on the toxigenic bacteria lecture, which you did for half a session. So he'd have 15 questions. Today's session, both lectures are mine. There'll be 10 questions from today's session. All right, so set up your studying accordingly. Like everything is of equal weight, even though some of the lectures might have been a little bit shorter than the others. You will have 90 minutes to complete the exam, so that's an hour and a half. It's about a minute and 45 seconds per question, which should be plenty. Last year, that's actually an increase. Last year we did, historically I've done about a minute and a half per question, but this year I decided that certainly for this first exam anyway, I'll give you a little bit longer because, this might be the first time you've seen these clinical vignette type questions, and those can sometimes take a little bit longer to pick through. So we'll do it in an hour and a half, and we can have a discussion as a class. After that, if you felt it was a little pressed for time, we could even increase that as we go forward in the course. So that's 90 minutes. Now, at the end of that, you will have an opportunity, this is done in ExamSoft, so you get your score right away, and you'll have an opportunity to review the exam. This is the only opportunity you'll get. Again, this is something new this year. Historically, in this course, we've never let the students review the exam, but ExamSoft does have a mechanism for doing this, and I'm going to try it out this year, and we'll see how it works. So, those instructions I sent you out last week, read them very carefully, because what's going to happen is when you finish your exam, you're going to be given an option to either upload the exam or proceed to the review. If you upload the exam, that's it. You cannot do the review. So take your time, be very careful in what selection you make at that point. So make sure you select, proceed to the review. And that will require a second password, which I will, you can just reach out to your proctor at that point and say, I'm finished. I want to do the review now. Can I have the password? And they will give you the password and then allow you to go to the review. Now the review, so you'll be doing that individually, right? Each of you will do it individually, and you'll be able to, basically see what you got right, what you got wrong. Most of you, I'm sure, will be interested in questions that you got wrong. And you'll have 30 minutes. We've done this in other courses, and I've never seen anybody take the full 30 minutes. Really, generally a student may spend 5 or 10 minutes on the review at the most. And then once you're finished your review, then you will upload the exam, and then you will get the green screen, and you'll have to show the green screen to the proctors. But please, I've posted the instructions online on the Canvas page. You have the instructions. Read them very, very carefully. Make sure you understand them before you come to the exam. In terms of the proctoring, you're going to be divided into groups of 20 each, maybe a little less, maybe 15 to 20. And and each group will have a minimum of 2 proctors associated with it. So we are going to be proctoring. We're not going to use this ExamSoft function where they have their own proctoring service. You may have used that in other courses. The problem with that is that it doesn't allow you to have a blank piece of paper, use a piece of paper. And I think that's really important to have those, blank pieces of paper. Most of the students really find that very, very helpful for the exams. So we're going to proctor it. What's going to happen is we're not, you won't be using this link do not use this link we're on now for the exam next week. In fact, the first thing you'll get probably, tomorrow is you'll see that next week's session is canceled. I will just simply cancel this link or this session for the course for next week. And then sometime before the end of the week, maybe even over the weekend, you will be invited by one of the faculty proctors. It could be myself, it could be Dr. Bellafato. There's a number of proctors involved. You will be invited to a Zoom meeting to do the exam. And that is the link that you will use. Now, remember, because we are proctoring this, and again, read the instructions carefully, you're going to need 2 devices, right? So you're going to need the device which you will be joining the Zoom session. That's probably going to be your phone, maybe. It's got to have a camera on it. So your phone maybe, or an iPad. And then you're going to have your computer, which you're going to do the exam on, which will be, you'll need Exemplify, obviously, that software loaded up. And I assume you all have the software. If you don't have the software, you really should reach out to the graduate school and they'll get you sorted out with that. Let me think, what else? Types of questions. I posted some representative questions. These questions are provided by the lecturers, so it gives you an idea of what kind of questions. You'll notice this is the course is content-based, right? So you learn the content and you'll learn better. Those students who know the content better, who review the material more thoroughly, learn the content more thoroughly, will do better on the exam for sure. There are, 2 kinds of questions. A lot are just, information that the different professors have provided to you. It's generally things they've talked about, almost certainly on a slide of it, and on a slide and something that was talked about. You will see, and as I say, you can see them on those practice questions I posted as well. There are some clinical vignettes involved as well. Those are, you'll see a lot of those, for example, associated with the material you're going to hear today. So those are a little bit different because you have to sort of pick your way through, some symptomology, some tests and things like that. But at their heart, they're just, it's information, right? It's content, right? It's just content that's been packaged in a different way. So don't waste time trying to find outside resources, outside question banks and things like that. They're going to be I guarantee you, they will be absolutely no use to you. That will not be high yield studying. High yield studying for this course and for these exams is learning the content. All right, and particularly focus on the videos, review the videos that we provided with you. Listen to what the faculty are talking about. It almost certainly will test you on something that they spoke about referring to a slide, or it may not even be on a slide, but if they've talked about it, there's your high yield studying right there. Now, I'll tell you what I'm going to expect from this exam. I'm going to expect a fairly low class average because the exams tend to be challenging and they're difficult. Last year, I think the class average on all of the exams was in the low 70s, anywhere from 72 to 75. you're going to be lower. So you guys, a lot of you are going to get lower scores than you're normally used to getting. That doesn't mean you're going to get a lower grade on the course than you're used to getting. I like difficult exams because it spreads out the class, and I can, you know, and it's easy then to makes it a little easier to separate, you know, the very, very strong students from the less strong students. So I told you, you know, the question, I will answer it, you know, do I curve? I absolutely curve this course, all right? I told you at the beginning, my matrix is over 90 is an A. That's where I start. I believe last year, an A for the course ended up being an 80, all right? And I think a B for the course ended up being a 70. And so I actually had to, scale the course down, I think by 10 percentage points from what I started with. And there's no reason why I wouldn't, or I expect maybe to be doing the same thing again, because I don't expect the exams to be any easier. So don't, you know, you're gonna see these scores you got on the exam before I'm able to talk to you about the scores. So don't get too anxious about the scores. As I say, if they're lower than you're used to seeing in other courses. What I'll tell you is, you know, when we meet the next time after that, you know, after the exam, I'll tell you what your score means. You know, if you got a 70, I'll tell you, you know, what that means, you know, roughly in terms of what that would equate to. If I were to end that course at that point in time, what that 70 would mean in terms of a letter grade and things like that. So I'll give you, and we're going to do this after every exam. I'll just, you know, try to give you guys a really good sense of where you stand in the course. Generally, something like the top 25% of the class gets A's in the course. So that's what you're shooting for. You're shooting for an A. I'm sure many of you are. You're really shooting to be in the top 25% of the class. Okay, I think that's. I think students have always found the grading over. As I say, they're difficult exams. You know, some students just nail them. You know, I mean, we do get every year we get a couple of perfect scores, but you know, many students find them. As I say, more challenging than they see in other courses. So what else? I don't see anything else, but questions. Is there any questions? Okay, good. Again, I emphasize, please read the instructions. Please, the exam starts at 2 o'clock. The same, you know, 2 o'clock next Tuesday, that's when it starts. Do not log on at 2 o'clock, all right? The exam will be starting at 2 o'clock, which means there's a procedure that you have to go through before the exam starts, right? So the proctor is going to take you through a whole series of, we're going to be looking at your workspace, right? We're going to be asking you to pan your device around the room. You need to be in a quiet place. You need to be by yourself. There can be nothing in front of you. We're going to want to see that piece of paper. We're going to want to see that it's blank on both sides, and you're going to be given some instructions, all right? And the idea is that we start the exam right at 2 o'clock. So, you know, that meeting will start, that you're invited to, will start at 1:30. Log on early. If you have trouble logging on, you know, we're going to start the exam at 2. There's going to be nobody available to help you log on at 2 o'clock. But if you're having trouble logging on at 1:30, that gives us some time to sort out some technical issues. So please don't be one of these, you know, 1:59 jumping on and, you know, everybody's sitting there ready to go. And then we have to start all over again and you know, and look at your thing. So your workspace and that. All right. So, you know, 1:45. You'll start at 1:30. If you come on at 1:30, that's great. You can just check your camera, make sure everything's working, you know, walk away, go have a sandwich, and then come back. But make sure you get logged on good and early, and make sure you're sitting in your chair at 1:45. You know, if everybody's on in their different meetings, we can, you know, your proctor may just even decide to start you guys a few minutes early, and you can finish up a bit earlier. There's a question, do we have to keep our mic on during the proctoring? No, during the proctoring, you're going to have your video on, everybody is muted, all right, including the proctor. So the proctor will be muted as well. This is like it's gonna be like a classroom. There's to be no sound whatsoever, all right? Remember, if you're having a problem, you need to talk to a proctor, work through the chat. You will not be able to chat with one another. That will be, that feature will not be available to you, but you'll be able to communicate with the chat with the proctor. Use the chat, all right? Because if you just turn off your mic, that would be like, you know, sitting in a classroom and just, you know, calling out a problem that you have while everybody else is doing the exam. And so, yeah, so the question is, you'll all be mute, your cameras will be on, you'll all be muted. The proctors will also be muted, and their cameras will be off, okay? But they will be there and they will be watching everybody very carefully. Any other questions?
Professor Whitehead?
Yeah.
I know you said this, I just had trouble hearing you. How many questions will be on the exam? Did you say 70?
No, 50, 50.
Okay, thank you so much.
Okay. Okay, great. And, you know, I'm going to lecture here for a couple hours, so we're going to be here for a while. If you think of any other questions, I'll be happy to stay on the end, and we'll and I can talk about the exam. Anything more you want to talk about the exam then. So let me just go ahead. Okay, everybody see that? Does somebody unmute and say yes?
Yes.
Yes. Yes.
Very good. All right, everybody hear me fine?
Yep.
Yes.
Okay, great. So let's get started. So what we're going to do today is we're going to now turn our attention. Up to now, we've been talking about bacteria in general. What we're going to do now is start a series of lectures where we're going to talk about the bacteria themselves, different types of bacteria, and the diseases that they are associated with. And we really focus the course on the most important ones, the most important pathogens. So, you know, for example, today I'm going to talk about pyogenic gram-positive bacteria. I'm not going to talk about every species of pyogenic gram-positive bacteria. I'm going to talk about the ones that are most common and the ones that are associated with the more severe diseases. Okay. So pyogenic. So what does that term mean? It's not pyrogenic. All right. So don't get the two terms mixed up. Pyrogenic would mean, you know, heat causing or things that cause fever. That's not what these are. These are pyogenic, and pyogenic means that they form pus. All right, so if the infection is a localized infection, maybe on the surface where you can actually see it, maybe the back of your throat or on your skin or something, that infection will lead to the formation of pus. And pus is a liquid, generally yellowish in color. It's going to contain bacteria. It's going to have dead bacteria. It's going to have live bacteria. It's going to have neutrophils, macrophages, you know, all these white blood cells that are trying to kill that bacteria. So all of the strains that we're going to talk about today share this feature. They form pus. All right. They're all gram-positive, and they are all parts of your normal microbiome. All right, so as all of you sitting here, they're watching these lectures today, all of the species that we're trying to talk about today, at least some of you will have these species in your microbiome. Now, most typically, you're going to see them in the skin and or in the nasopharynx. All right, so pretty much all of them could be found in one of those two locations. Now, that doesn't mean they're in both. All right, they can be in both, But let's say, you know, I have one of these strains on me. Chances are it's either on my skin or it's in my nasopharynx. It's generally one or the other. You know, more seldom you might have it in both locations. It also doesn't mean that if I have it on my skin today, I'm going to have it on my skin next week. So again, all of you listening to this lecture today, I would say probably about 25% of you probably have Staph aureus on your skin right now. And if we came back here, met back here 5 years from now, I'd say the same thing. 25% of you have Staph aureus on your skin right now, but it wouldn't necessarily be the same 25%. All right, so we have these things, they go away, we get reinfected with them again, they go away again. Right? So that's, you know, that's kind of the general sense of their distribution. And for the most part, they're on your skin. They're not going to cause any disease at all. As I say, they're part of the normal microbiome, but there are circumstances, things that can happen, maybe a cut in your skin, you become immunocompromised, you know, that can, and your, you know, bacterial defenses might be dropped. Your antibacterial defenses may be lowered for a variety of reasons. And sometimes we don't even know why they infect it, but, you know, on occasion they will infect, and some of the strains associated with them certainly can cause disease. All right, so remember, these are all, everything we'll talk about today is part of your normal microbiome. Those are just my objectives. Yeah, exam next week, so you should start, you know, as you start to prepare, learning this material, start with these objectives. This is what I'm going to hold you responsible for. All right, so we're going to talk about two genera. In the first half, we'll talk about the Staphylococci, and in the second half, we'll take a break and then we'll talk about the Streptococci. Now, although I say we're going to talk about this genera as a Staphylococci, the reality is I'm going to spend 99% of this first period talking about one species, and that's Staph aureus. And the reason for that is it's the most highly infectious of all the Staphylococci, and it is responsible for the most disease, a huge range of diseases, as you'll see. The second hour, we'll talk about Streptococci, and we'll talk about a few more. I think we're going to talk about 6 different strains, but even there, one of them, Strep pyogenes, again, like Staph aureus, extremely infectious, associated with a broad range of disease. So we'll spend at least half of that second session on pyogenes, but there are others that are, you know, I would say they're equally serious if you get infections, but they're somewhat less common, but we'll touch on those as well. All right. So but everything we're going to talk about today, gram-positive cocci, they're all, I say, in the nasopharynx, or you'll find them on your skin. I put GI up here as well on this slide because, as you'll see, quite a few of them, but not all of them, can also be found in your gastrointestinal tract. Some you'll see in the genitourinary tract as well. So they're not limited to these particular niches. There are other niches that they can inhabit. Right away, you can see one difference between these, the two genera, and that's how they grow. So if you took staph, you took a culture, grew it in liquid, grew it overnight in a liquid, came back the next day, took some of that liquid, put it on a slide, take a look at it under the microscope, you would see it grows in these clusters, these grape-like clusters. Streptococci, you do the same thing. You don't get those grape-like clusters. What you'll see is one of two things. You can see what are called diplococci, which are just pairs like this. Good example of this is Streptococcus pneumoniae, which we'll talk about later. So that's the only, actually, I think that's the only strain we're going to talk about today, or the only species that grows like this. But the rest of them would grow like this in these chains. All right. I will tell everybody that I don't have anybody looking at the chat today. It's difficult. It's very distracting for me to monitor the chat and lecture at the same time. So if anybody does have any questions, please just feel free to unmute and just jump right in with your questions. All right, that'll be fine. Now, as I'm talking about these different strains today, I'll not only describe them to you, the strains to you, and I'll describe the diseases associated with them, but I'm also going to describe to you how to distinguish between them. And as you can see by those practice questions I put up and the objectives for the lectures today, it is important that you be able to distinguish between them. And the reason for that is that they're often found in the same niches, like, staph and strep are both found on the skin. As you'll see, they're often associated with the same diseases, right? So, it's on your skin, it's causing impetigo, which we'll talk about later. That doesn't really tell you, it could be staph or strep, but you need to be able to figure out which one it is. And the reason for that is because of different antibiotic resistances associated with different genera. You're going to treat them differently. So you're going to treat a staph infection probably different than you might treat a strep infection. And so you have to be able to distinguish. So in addition to telling you about all these different species today, I'm also going to tell you the tests that you can do to distinguish between them or to figure out exactly what species that you're dealing with. And it's really important that you know what these tests are and how they're used and what these tests tell you, because I guarantee you it's going to show up on the exam next week. Your ability to do this is going to show up on the exam next week. And even as we go further into the course, more and more, you're going to see questions where you're given, the information in the question might be the results of a test. And you're going to need to know what the meaning of that result is with regard to the identity of a particular strain of virus or a bacteria or yeast or, whatever we're covering. Right? So here's our first test. Now, I already told you one way to distinguish between staph and strep is to, grow these cultures overnight, and staph grows like grape-like clusters, strep grows a little different. I wouldn't exactly call that a highly reliable test, and it's certainly not something that really could be automated. And the better tests are the ones that are automated. So I'm going to give you other tests as well. So I'll probably give you several tests which can be used to distinguish between species. There, fortunately, is a very nice, easy, straightforward test that allows you to distinguish between a staph infection and a strep infection. And that's the catalase test. And that's because staph produces catalase and strep does not produce catalase. So it lacks catalase. And the reaction this is the reaction of the catalase test here. All right, so you start with hydrogen peroxide, you add catalase to hydrogen peroxide, that will convert it into water and oxygen. And what you're going to be looking for is the production of the oxygen, because the oxygen that's produced is going to be a gas. So let me see if I can, here we go. So if we have staphylococci, for example, we can take a slide, we'll put some hydrogen peroxide on that slide. We'll take the staphylococci itself, some of the bacteria on a swab, we'll rub it into that hydrogen peroxide. And if it's producing catalase, it'll react with the hydrogen peroxide, it'll produce oxygen, and you'll get bubbles forming. And it's very, very obvious. Right? Do the same thing with streptococci, doesn't produce the catalase, you don't get the reaction, you get no bubbling. And this is something, in this case, you could actually see it. I've done these tests and you can see them. Now it's automated and it just measures the production of the gas. All right, so that's an important test that allows you to distinguish, tell what kind of infection you have. All right, now let's turn our attention to the staphylococci, the species. In common properties, gram-positive cocci, normal flora, commonly in the nasopharynx, which is basically at the back of your throat, and on your skin. Something I haven't talked about about the staphylococci, but again, it's a little different than strep. They're extremely resistant to drying and heat, and they're actually resistant to many antibiotics, which is why it's important to know whether you have a staph infection or not. But this resistance to drying and heat, what does that mean? Well, what that means is that, remember, you can have staph on your skin. So staph could be on your hand. You could walk through a door. So you grab that doorknob as you walk through that door. And if you have staph on your hand, you'll transfer some of that staph onto that doorknob as you leave. Right? Now, what happens to bacteria? If you put bacteria on a surface like a doorknob, the vast majority of bacteria are not particularly resistant to heat or to drying, and they will simply die. And they may die within seconds or within minutes. Right? Something like staph, however, it won't die. It'll stick around. It can live on that doorknob. Not necessarily, it won't grow, but it'll just stay there, not dying. And that can be for 5 or 6 hours. Now you got a whole stream of people walking through that door. Every time somebody walks through that door, that staph will be transferred onto their hand. And that's why these species are part of the microbiomes. And sometimes we have them, sometimes we don't. They move around very easily from individual to individual. And if everybody's very healthy and this door is just in a classroom, and that's probably not such a big deal. Where it becomes a big deal is if this is occurring in a hospital. All right. And the reason it's a problem in a hospital because, as you'll see, it's not like every strain of Staphylococcus is going to cause disease. Some strains don't cause disease, even if you're talking about aureus, right? There are strains of aureus that don't cause disease. There are some that do. Most of the strains you encounter in your daily life and that you're picking up don't cause disease. Where, however, are you likely to find strains that do cause disease? And that's in hospitals, right? Because people are in the hospitals because they've got the disease from the bacteria. So they're carrying the disease-causing strains. And now, so they walk through a door and touch a doorknob. Now you've got a disease-causing strain that now can be passed to other people. And so that's, as I say, a major problem. The second issue with hospitals is there are a lot of unhealthy people. That means there are a lot of people who are immunocompromised, people who may have burns, people who may have wounds. So when they get these disease-causing bacteria on their skin or they breathe them into their nasopharynx, those are then able to very easily invade into those individuals and cause disease. So these are a major problem with nosocomial infections in the United States. So huge, huge clinical problem. All right, so we're gonna, as I say, I'm gonna spend most of my time talking about Staph aureus. I'll only mention one other strain, and that's Staph epidermidis, and we'll just talk about that briefly. I think this is the only slide in which I mention it, but you are responsible for it. Staph aureus, as I said, highly infectious. And the reason we're going to spend so much time is because of this figure over on the right here. And these are all the different types of infections and diseases that can be associated with different strains of aureus. Very common, probably most common, are skin infections. So skin, soft tissue infections, and we'll sort these just different types of infections you can get. And we'll talk about those as a little later. Much less common are musculoskeletal infections, but you can get them. This can lead to things like arthritis. We're not going to talk about this today, but again, just be aware. It's another, this is kind of disease that can be associated with a staph infection. Endocarditis, we'll talk about that a little bit. There are a lot of different species that we're going to talk about today that can cause endocarditis. Endocarditis is infection of your heart muscle, particularly the cells around your heart valves. It can cause inflammation of the heart and pretty severe heart disease. And as you'll see, even epidermidis can cause endocarditis. And again, that's why it's important to be able to know what you've been infected with. So we'll talk about that. Genital urinary tract, again, you can associate with aureus, much less common, but you can get infections. We won't touch too much on them. And toxins. So Staph aureus, there are different strains that will produce toxins. There's 3 different diseases that are caused by those toxins that we're going to talk about listed here, and we will get to them. Epidermidis, as the name implies, epidermidis, right? That's comes from epidermis. This is by far the most common, the most dense bacteria you're going to find on your skin. You'll find more Staph epidermidis on your skin than any other bacterial species. That being said, it is an extremely wimpy species when it comes to infection. So, you know, infections with it are relatively rare. Unlike Staph aureus, which is present at much smaller amounts and can infect, you know, much more efficiently, epidermidis has a hard time getting in. In fact, such a hard time getting in that the only time that we really see infections associated with it is when an individual might go in for an operation, and that operation generally involves a break in the skin, right? So a lesion in the skin, and you're going to put in some kind of implanted device. So maybe you've got somebody come in where you're going to put a pacemaker in, perhaps some kind of stent in them. And what will happen is, as these pacemakers, these devices pass through the skin, they'll pick up epidermidis on them. And so as you put the device in, you're actually implanting the bacteria into them as possible. And, you know, obviously, you know, you try to have as sterile technique as you can, but when the whole body is covered with this bacteria, it's very difficult sometimes to keep it out because of that. Now, if it gets in, it can get into your blood, and that's going to cause what's called bacteremia. Bacteria gets into your blood, it'll start circulating. And one of the places epidermidis likes to go is to the heart. And it'll infect those cells of the heart around your heart valves, cause them to become inflamed. And that is endocarditis, which you'll see is, you know, is something we're going to see more of as we talk through these bacteria today. Because both aureus and epidermidis can cause endocarditis, right? So it's coming up on both here. Again, it's important to be able to distinguish between the two of them. So here's a second test. Here's another test you need to know. Remember, both of these are catalase positive because they're Staph. So that's, you know, distinguishes them from being strep, but it doesn't distinguish them from one another. So in this case, the test is called a coagulase test, another convenient test. Aureus is coagulase positive, meaning it produces coagulase. Epidermidis does not produce the coagulase. In this case, this is the reaction here. The reaction is you start with fibrinogen, which is a soluble molecule. You add coagulase and prothrombin, and you will cause the fibrinogen to be converted into fibrin, which is a clot. So it's a clotting reaction. And again, this is something that can be automated. So what you do with the test is you'd have a test tube with the fibrinogen in it, you would add the prothrombin, and then you'd add the bacteria, either aureus or epidermidis. If it's aureus, the aureus will produce the coagulase, and what you'll see is clotting taking place in the test tube. If you add epidermidis, you see no clotting take place, and the fibrin doesn't form. Again, it's an easy test. You don't even, you know, it's easy to see in a test tube, but again, it's a test that can easily be automated. All right, so let's start to focus and hone in now down on Staph aureus itself. So Staph aureus has what we call a very high pathogenic capacity. And a high pathogenic capacity means it's a very powerful pathogen, and it can infect very easily, and it can cause disease very easily. That's, you know, opposed to epidermidis, which has a very low pathogenic capacity. And the difference between them is, you know, strains that have a high pathogenic capacity, things like aureus, have a lot of surface molecules, a lot of factors that they secrete, and these things combined will allow the bacteria to successfully, efficiently infect. And often they have toxins, they will secrete toxins, and they can be different types of toxins, and they will cause different types of diseases. So I'm going to just give you some examples of these for aureus. I think we'll go through a total of 6 or 7 different examples here that, you're responsible for all of them, but as you're memorizing them all and, grumbling about it, remember one thing: we're only scratching the surface here, okay? If you're in dental school and doing this course, if you're in medical school and doing this course, we talk more about 12 to 15 of these different factors. And again, it's important to know them. And that's what the medical students are expected to know. If you were to then go on and, and for example, and be a clinician and specializing in infectious disease, aureus has over 100 of these factors, and you would need to know all or learn all 100 of them. So we're going to try to keep it as limited as possible, so you don't get overwhelmed with these things. We'll just talk about a few of the more interesting factors. But as I say, just keep in mind we're only, we're just only scratching the surface here of what this bacteria can actually really do during an infection. So let's begin by talking about a couple of interesting surface molecules. And these things I don't think you've heard about fibronectin binding protein, but protein A, I think you've already heard about, because I'm pretty sure Dr. Beaulieu talks about this in her lecture. So fibronectin binding protein, as the name implies, is something that binds to fibronectin, and it serves as an adhesant. So here's the staphylococcus, and your fibronectin binding protein is going to be here, and it's going to be on the surface of the bacteria, and it will allow the bacteria to bind to anything that has fibronectin on its surface. So what has fibronectin on its surface? Well, mucosal cells do, and tissue matrices do. So remember, staph not only infects skin, it affects your nasopharyngeal pathway or passage as well. That passage, that's covered in mucus. So underlying that mucus are mucosal cells. They're epithelial cells, and they express fibronectin on the surface. So when Staph aureus, if you breathe it in and it gets into that mucus, it can then find that fibronectin on the surface of the cell and bind to those cells. And it needs to do that. It needs to adhere to those in order for it to then translocate between those cells into the underlying tissue matrix. And when it gets to the underlying tissue matrix, it'll find more fibronectin, and that'll be part of that tissue matrix itself, the extracellular matrix, and it'll bind to that as well. And that binding is important for the bacteria to then start replicating. So that's one factor. Another factor is protein A. And again, I think this is something you've heard about already in a previous lecture. So protein A, again, is a protein shown here, is expressed on the surface of the bacteria. It also binds to something, and the thing it binds to is the Fc arm of immunoglobulins, IgGs, actually IG1, IG2, and IG4. Now, you might be asking yourself, if you haven't, been reviewing this material yet, why would, the bacteria binding to antibodies help the bacteria? Wouldn't that just target the bacteria to be engulfed by a macrophage or a neutrophil? Right? Well, let's, take a look at exactly what's taking place here. So here's what normally happens. So here we have a normal bacteria. And here we have antibodies that are binding to that bacteria. There's the Fc portion, the long part. Remember, antibodies are shaped like Ys. They have the long arm, the Fc portion, and then they have the 2 shorter arms. And what will happen is it's these shorter arms that typically would bind to antigens on the surface of the bacteria. They would coat that bacteria, get an opsonization reaction, and then those would be recognized again by a macrophage or a neutrophil, and that whole complex would be engulfed. Now, if that bacteria, however, is expressing protein A on its surface, what happens is that protein A still will actually binds to the antibodies, but it binds to the Fc portion, to the longer arm. So what happens here is it flips the orientation of the antibodies. So now the short arms are facing outwards, and you'll get an opsonization opsonization, it'll be completely coated. And this is very non-specific binding. Any antibody now, any IgG antibody will now bind to this bacteria, but it actually is going to protect the bacteria because the antibodies in this orientation are invisible to macrophages and neutrophils. They don't recognize this conformation and they can't engulf the bacteria. So the bacteria is coating itself with antibodies, and in doing so, it's protecting itself against the host immune response. You know, very, very clever. Let's talk about extracellular factors. Now I'm talking about things not that are expressed on the surface. I'm talking about things that the bacteria actually secrete. And there are things that it can secrete that, again, will support infection. And there are things that the bacteria will secrete which seem to be more involved in causing disease than actually supporting the infection. And why they cause disease, and in some cases, killing the organism, killing the host, is anyone's guess, but we'll talk about them. First, let's talk about an extracellular factor, something that's secreted, doesn't cause disease per se, but helps the bacteria, again, be successful. And these are the hemolysins, all right? And hemolysins is a general term. There's many bacteria that can produce hemolysins. The one that Staph aureus produces is called alpha toxin. Again, this is something you've heard about in previous lecture. Alpha toxin is a toxin that's produced that creates transmembrane channels. So the bacteria secretes it, they insert into the membrane of cells that are mammalian cells surrounding the bacteria. They create these channels. You get water flowing into the bacteria. Those cells become under osmotic shock, and those cells will simply explode or lyse. So these toxins will actually lyse mammalian cells. And we know that this alpha toxin that Staph makes can actually lyse both red blood cells and white blood cells. The white blood cells we understand, I'll talk about that in a second, and that's, has a clear role in infectivity. The red blood cell thing, we're not quite so sure whether this is really something that's going on in vivo or not. It's something that we know that the hemolysins can do. We just don't know whether the bacteria is really doing this to any great degree. And the reason for that is we don't really see in these infections, although they're producing the alpha toxins, we don't see a lot of anemia associated with these infections. So they are killing red blood cells, they're not doing it a lot. That being said, we can use it for diagnosis. It can be helpful in diagnosis. So many of the bacteria I'm going to talk about today, Staph aureus, Staph pyogenes next. We can grow them on regular agar plates, but sometimes we'll grow them on enriched plates. And one of the types of plates that they're often grown on is blood agar plates. And a blood agar plate is a normal agar plate that's been enriched with a lot of different nutrients, as well as red blood cells. And generally, these are sheep red blood cells. So the plates are actually red. Okay? If you grow your Staph aureus on that plate, what will happen is they'll get nice colonies forming on the plate, and then those colonies will secrete the hemolysins. Those hemolysins will then lyse all the red blood cells that surround that bacterial colony, and you'll get what's called a zone of clearing. It's called beta hemolysis. And we'll talk about, and I'll actually show you what that looks like a little later when we get to pyogenes later. So you can actually see this lysis taking place when the bacteria is growing on these, but again, just on these, what we call blood agar plates. Again, does that have anything to do with infectivity? Maybe, maybe not. Red blood cells are a good source of iron. Bacteria do require iron to divide. So it could be that that aureus is lysing some red blood cells to scavenge the iron from them and using that iron then to support replication. That's sort of a theory, but there's no hard proof that that's actually taking place. White blood cells, however, this really is important for virulence because what's going to happen, as I say, is these bacteria, they're going to try to protect themselves from opsonization, and some will be successful, others will be less successful. If they're not successful and they're properly opsonized, they would be targeted by neutrophils and macrophages. But this is now a second line of defense. The bacteria can then, produce these hemolysins, and they will get in the membranes of these white blood cells and lyse them. And that truly does happen, and that truly does help protect the bacteria. Now let's talk about the toxins. All right. So now we're talking about more factors that are actually secreted. They're extracellular factors. They're actually secreted, but we're not really sure whether or not they're helping the infection per se. But what we are sure about is that they are causing disease. And there are different exotoxins that different strains of aureus can make that can cause different diseases. So I'm going to tell you about 3 different toxins. Now, that doesn't mean that all aureus all strains of aureus make toxins. That's not true. It's actually a very limited number of strains that make these toxins. And just because they have the gene for the toxins doesn't even necessarily mean that they express the toxins. Right? They may only express those toxins under very unusual or limited circumstances. But if you do get infected by one of these strains that has the gene, and the circumstances are right, are perfect for that toxin for being produced, and the bacteria does produce that toxin inside you, it can cause some pretty serious diseases, as you'll see. All right, so we'll talk about 3 different toxins. These are all called exotoxins. First one we'll talk about is TSST1. That is, again, if you have a strain that makes this toxin, it can be, if the toxin is produced, associated with toxic shock syndrome. TSST1 stands for toxic shock syndrome toxin. All right, toxic shock syndrome toxin 1. And it's an antigen, a super antigen, and 20% of S. aureus strains carry this gene. So 20% are capable of producing this toxin. Now, again, just because you have the gene doesn't mean you necessarily produce the toxin. It can only be produced, as you'll see, under very unusual and rare circumstances. But let's say it's there in a lot of strain. You've heard about superantigens in previous lectures. I think you might even, both Dr. Hermione and Dr. Brullier may have talked about superantigens. So I'll just go through this very, very briefly. Right, just to remind you about what a superantigen does, because I'm going to talk about a couple more superantigens later this afternoon. And so here's our normal circumstance. Here's our antigen presenting cell. And on its surface, it has these antigen presenting cell receptors, or APC receptors. And an antigen will be recognized, and the antigen will be inside the antigen presenting cell, and that antigen will be presented on, and that might be a surface molecule from a bacteria, for example, and that will be presented in the cleft on that receptor on the surface of the antigen presenting cell, and you will get T cells, which have T cell receptors circulating, and they're sampling all these antigen presenting cells, looking for an antigen that they might recognize. And for any given antigen that an APC presents, maybe 1 in 10,000 T cells, circulating T cells, will actually recognize it. They'll recognize it, they'll bind to it, cytokines will be secreted. And this is not an immunology course, so I'm not requiring you to know a lot of immunology here, but there's a lot. You're going to get a robust immunological reaction. That T cell will start proliferating. It'll interact with a B cell. The B cell will start proliferating. It'll start producing the antibodies, which can then target, the bacteria that actually has that antigen on it. And this is sort of normally what happens. And the important piece to this whole thing, the thing you have to remember, is this here. It's highly selective interaction. So only 1 in 10,000 circulating T cells will recognize any specific antigen. Now, a super antigen is something, again, this is a molecule that's being secreted by Staph aureus. Okay, so it's in circulation. There it is there. It never actually ever makes it inside the antigen presenting cell. But what will happen is as it goes by and encounters an antigen presenting cell, it will bind to the APC receptor. And it binds to the APC receptor on the outside of the receptor. It'll actually prevent an antigen from actually even binding into that cleft. So it's actually just on the outside of the receptor. And the other end of that super antigen will now bind to T cell receptor. But the important thing is this is a non-specific interaction. So now instead of having 1 in 10,000 T cells binding, as the T cells come by and sample, you're going to see as many as 1 in 5 T cells binding to this antigen. They are now going to be stimulated, now going to get a huge amount of IL-2, TNF, all these cytokines are going to be produced. You're going to have what's called a cytokine storm take place, and that's going to lead to shock. And we'll talk about that disease in a few minutes. Right now, I'm just going to talk about the mechanisms. We'll talk about disease, as I say, a little bit later in the lecture. So that's one exotoxin. Here's another one that's Staph aureus. Now, again, any given strain isn't going to have all of these genes. So you might have a strain that produces, TSSD1, but it's not going to produce the exfoliative toxin. So you might have another strain that produces an exfoliative toxin. So exfoliative toxins, there's actually 2 of them, A and B. You don't have to remember that. Just remember exfoliative toxins. They both do the same thing. And they cause a disease we call scalded skin syndrome. We'll get to that in a few minutes. But let's just talk about the mechanism, what these toxins actually do. So these toxins, A and B, are actually proteases. So they cleave proteins. And the proteins they cleave are the desmosomal proteins of the skin. So here is a cross section through the epidermis. So this is the outer layer of the skin, showing the different layers. There's the stratum corneum, stratum granulosum, spinosum, basal. You don't have to memorize these names here. Okay. Now, notice that these different layers of the epidermis, these different layers of cells, are actually being held together. And they form a nice barrier. And the proteins that they're being held together by are called desmosomal proteins. And there's different types of desmosomal proteins. So this one shown in blue that you find more commonly in the stratum granulosum is called DSG1. This is another one shown in yellow, which you see more commonly down here. That's DSG3. So what these exfoliative toxins will actually do is cleave these desmosomal proteins, but they're very specific. They're specific for DSG1. So they get into the stratum granulosum, into this layer here. They cleave those desmosomal proteins. Those cells float apart, and that allows the outer surface of your skin, the stratum corneum, to sort of slough off the surface. And that's called scalded syndrome. And we'll talk about it, that disease, a little bit later. All right, we'll get back to this figure when we talk about the disease in a few slides. The third enterotoxin that staph can make, or some strains can make, is one that causes food poisoning. Right? And it's also a super antigen. Notice I'm just mentioning it here. I'm not talking about, we're going to talk about the food poisoning in a few minutes, but I'm not going to talk about the mechanism. And the reason is we don't know the mechanism. We know it's a super antigen, but it really never gets into the bloodstream. Now, a super antigen becomes a problem if the toxin gets inside you. This particular toxin never gets inside you. It stays in your gastrointestinal tract. So although it could function as a super antigen, that's probably not the mechanism by which it causes disease. But we don't know the mechanism, but we'll get to the disease in a minute. Oh, here we are. Now we're going to start talking about staph infections and the diseases. Back to this, I already sort of went through all the different kinds of diseases that staph infections can cause. Now I want to go, as I say, through some of those diseases, the more common ones, and we'll just talk about how these, what a patient might look like if they had a staph infection. We'll talk about the skin and soft tissue infections. These are by far the most common types of staph infections that you might get. We're not going to talk about musculoskeletal because they're so rare. I've actually already mentioned endocarditis. I might just talk about it briefly again. We're not going to talk about genital urinary tract infections, but these are the more serious infections, the skin infections here, and these toxin-based diseases that we've talked about the toxins. Now we'll talk about these diseases. So I'd say just keep in mind, I'm not going to tell you everything that staph can do to you, because it's a lot of things that staph can do to you. I'm just going to talk about the more serious, more common, more interesting things. Let's start by looking at, as I say, the most common type of staph infection which you will get, which will be a skin infection. These, we call them the suppurative infections. Suppurative is another word for pyogenic. It means they form pus. So these are infections that are associated with pus. And the reason they're associated with pus is because they're on your skin, so you can actually see the pus. I mean, if you had an infection on your heart, endocarditis, they might be producing pus there, but you'll never see it, right? Because it's on your heart. But these, you can actually see the pus forming on the surface of your skin. Now, let's take a look at this. This is again a cross section through the skin. We've already seen a cross section actually through the epidermis. Now we're going deeper, and we're seeing lots of different layers. And the reason I'm showing you these different layers is because the type of skin infection you can get, the type of disease you will get, will be dependent on which of these layers the infection gets into, where the bacteria gets to. All right, so on this side, we have the different layers. On this side are different diseases, some of which are associated, I shouldn't say diseases, well, they are, but they're, types of infections, skin infections associated with Staph aureus. And it's associated with some of these, but not all of these. And we'll see this diagram later when we get to pyogenes in the second session, because it can also cause skin infections. So let's say the infection just gets into the epidermis, this upper layer here. So the infection gets in, the aureus gets in, and it starts to spread laterally, and you'll get something called impetigo. Impetigo starts out as a pus-filled epidermal blister. So it's a blister that eventually is going to break and leave a thick yellowish crust. These are sometimes referred to as honey crusted lesions. I actually think there might have been a question on the practice questions that I gave you, where one of the involved a child who had honey crusted lesions on their face and behind their ears. And that's where you'll find these lesions, on the face and behind their ears. And that's generally associated with either a Staph aureus infection, or, as you'll see later, it could also be a strep pyogenes, produces exactly the same kind of infection. Excuse me. All right, so that's called impetigo. And this is actually what it looks like here. So here is a child, probably this kid is maybe 5, 6 years old. This blister is formed on his face, just underneath the nose. It's already broken, and you can see sort of this glistening pus. And that's on the surface of your skin, and that's just filled with bacteria. So that is highly, highly infectious, right? So impetigo is very, very infectious. Now let's say, okay, so here's our cross-section through our skin. Now, on our skin, we have hair. So in the hairs, here's a hair here, and it's sitting right within a hair follicle. let's say the infection, instead of getting into the epidermis itself, the bacteria makes its way into the follicle, stays still relatively high in the follicle towards the top, towards the surface, but again now starts to replicate within the follicle. That's referred to as folliculitis. And folliculitis, again, is something that is associated with a staph infection. It will, you'll get it will spread laterally from the follicle. You'll get an area of inflammation. It'll actually end up looking very much like a pimple initially, but it'll be more red. It's hard to actually distinguish from a pimple, but it's, let's say you'll get this pus filled and it'll be sort of very reddish inflamed area around it. Now, if that bacteria gets deeper into that follicle, okay, now getting down more into the dermis layer down here, in some circumstances, in some strains, these will start to proliferate very, very rapidly. So you get a tremendous amount of bacteria being produced, and they will produce almost like a ball of a colony of bacteria. And that colony will start to secrete materials that will form a capsule around that colony, and it will recruit materials from the extracellular matrix to support the formation of this capsule. So you get this big sort of capsule forming, which hard surface to it that's just filled with bacteria, and it can just erupt off the surface of the skin. And if that happens, that's called a furuncle or a boil, right? So that's how a boil forms, within a hair follicle. It's a more serious type of folliculitis. They're very hot, they're tender. I've never had one, my kids have never had them. So, I don't have any firsthand experience with boils, but my understanding is that they can be very, very painful. They can be hard to treat, and they can actually cause fever. The reason they're hard to treat is because of these capsules that surround them. So you have to lance the boil, which means you have to open up the capsule and you flood it with antibiotic to try and just, really just to kill all the bacteria in the boil. And sometimes that's successful and sometimes it's not. And if it's not successful, it might come back. And so again, people that have boils tell me that, sometimes they lance a boil, they hope they get all the bacteria, but when they don't get it, the boil can sometimes come back, in which case you actually have to remove the whole boil itself. So not just lance it, not just flooding it, but actually trying to sort of dig out and remove the whole capsule. And that can be quite painful. So they're hard to treat, boils. And so that's what a furuncle is. That's the medical term for boil. A carbuncle is a confluence of boils. So this is where you have more than one boil all clustered together. So here's a particularly bad case here. Just looking at the back of somebody's head, this is their hairline. And this is what happened is this individual probably had to start out with a single boil. They're probably just scratching it constantly, right? And remember, this is hair follicles. There's a lot of hair follicles here. So this just got into a lot of different hair follicles with this particular strain that causes boils. Get a lot of boils forming, and obviously that's going to be even more difficult to treat. Where else you might get pus forming infections with Staph aureus, if you have a surgery, so you have a wound, sometimes they'll form pus. Burns, I'm sure some of you are aware have had very bad burns. Staph can infect the burn. If it does, you can end up with sort of a yellow pus forming on the burn. But things can get worse. This can, if you have a deep wound or a worm, a burn, aureus can get in sometimes into your bloodstream. If it reproduces to a high enough level, it'll get into your bloodstream, and it will go to your heart. If you get bacteremia, even with the aureus, it will go to your heart, and it can cause endocarditis. Pneumonia. Yes.
I have a question about this previous slide.
Yep.
Is folliculitis a scientific term for like an ingrown hair, or is that different?
No, folliculitis is a scientific term for an infection of a hair follicle.
Okay, and that's different than an ingrown hair?
Yes.
So Is it an ingrown hair isn't an infection? It's just like, how does that?
Not really. I think it's just an issue with the hair itself. I mean, I think where the hair. I'm actually not even sure what
like an ingrown hair like has like, it kind of does have like skin over it. So that's what I'm thinking. Like there's a capsule over it, and
yeah, but it is. I'm sure it's different than folliculitis.
Okay.
Okay.
Okay. Thank you.
I just don't know the pathology of an ingrown hair. I'm pretty sure it's not the same as folliculitis.
Okay. Cool.
All right.
Thank you.
Professor, I have a question.
But I could be wrong, and if I'm wrong, I'll get back to you on it. I'm interested now, so I'll look it up.
Yeah, that would be great. Thanks so much.
Yeah.
Professor, I also have a question about the previous slide.
Yep.
So would a boil be a cyst, or are they two separate stuff? So
a cyst is a more generic term. A cyst is something that's used to describe a lot of things. It's just a cyst is something with a hard cover and a liquid in the middle. So that's, yes, a boil in many is a type of a cyst.
Okay, but a boil would definitely have bacteria in it.
Yes.
Okay, thank you.
By definition, it'll have Staph aureus in it.
Okay.
Okay. All right. Pneumonia. I'm not. Maybe 1 to 5% of cases of. Remember, aureus not only gets on your skin, aureus can also get into your nasopharyngeal region. So it can infect the back of your throat. And in rare cases, it can get down into your lungs and cause pneumonia. And that's generally associated with somebody who has the flu. So somebody who has the flu and has a lot of inflammation in the back of the throat, that can allow, some Staph aureus strains to then infect. Once they infect, they'll make their way down into your lungs and you can get pneumonia. It's rare. I mean, I'm going to talk later about strep pneumoniae that's responsible about 50% of cases of pneumonia in the United States every year. That's a staph infection leading to pneumonia. Again, it can happen, but very, very low, maybe 1%. I think 5% would be high. Maybe 1 to 2% of cases of pneumonia in the United States every year will be associated with Staph aureus infections. But just, be aware it's possible. And so, again, that just sort of highlights why it's important to be able to have these tests to distinguish, you have the same disease caused by different bacteria, and those bacteria need to be treated in different ways. So it's critically important to be able to figure out what exactly it is that is giving you the pneumonia. The toxic infections, scalded skin syndrome. So we talked about this earlier. Remember, it's the exfoliative toxins. What happens is it affects children, generally children under the ages of 5. It'll start with a localized infection, maybe something like impetigo. So in this case here, this child got impetigo, so it's localized. So they don't have bacteremia at all. There's no bacteria in their bloodstream. They've got this infection under their nose here. So they've got the pus forming there. And what happened is they've been infected with a strain, causes impetigo, but the strain is also producing one of these exfoliative toxins. And that toxin gets into their blood. The toxin itself, not the bacteria, spreads throughout their body, and it'll make its way into their skin. And if it gets into the skin, it can encounter these desmosomal proteins. It will cleave them, and it'll cause the skin to start sloughing off the surface of their body. It looks like they've been badly burned and are recovering a bad burn. Actually, if you've ever had a very bad sunburn, right? It's just the same kind of look. If you had a sunburn and, several days later you start peeling your skin, that's what it looks like. And it's about as serious as that. It doesn't really kill these kids at all. But, if you see a kid like this in the middle of summertime and they've just been at the beach for a week, you say, somebody was irresponsible and didn't put, sunscreen on this child. If you see a child who looks like this and it's the middle of winter and the sun hasn't been out for, a month, you have to think about, this kind of infection, that they may have a Staph aureus infection, and with this, what we call scalded syndrome. Diagnosis, it's actually hard to find, because remember, there's no bacteria here. It's really just your clinical findings. It's like, here's a kid who looks like they've got scalded skin, and it's the middle of winter and they weren't burned in any way. So that probably tells you what they got. And, the parent may say, well, it started out about a week ago. They had these, yellow crusted, scabs on their face, and then suddenly this happened. So that's what I mean by clinical findings. But it's very, you can't take this material and culture it and look for the bacteria, because there's no bacteria here. You might be able to go back up here to the nose and find the bacteria, but you're not going to find it down here. Toxic shock syndrome, TSST1, as I said, 20% of S. aureate. You're going to get elevated interleukin, TNF alpha levels. Remember, this is the super antigen. You're going to have a cytokine storm in your body occurring, massive amounts of cytokines. That's going to lead you into shock and all kinds of clinical symptom features, fever, rash, vomiting. And this can lead to multi-organ failure. But as bad as this all sounds, the mortality rate is actually relatively low. It's actually only about 3%. We're going to talk about things that are much more deadly than toxic shock syndrome. Again, when we get later to pyogenes, it has a very similar disease with a much higher mortality rate associated with it. So you're going to be really, really sick as a dog, but most people get better. Supportive measures, obviously antibiotics. You probably, if you suspect that your patient has toxic shock syndrome, you're not going to wait a couple of days to culture the bacteria and go through all these different tests. If you suspect, this person may, and there's different reasons this syndrome can form, but if you suspect that the person has toxic shock syndrome, you'll hit them hard and quickly with very heavy doses of antibiotics. It's actually very rare. You see it equally in men and women, although it was in the news a lot. I think it was back in the 1980s that this syndrome really hit the news, and we saw a lot of cases in women, and this correlated with the marketing of these new super absorbent tampons. So a number of different companies were producing these tampons in the 1980s. They were marketed as this was a tampon that only they were thicker, they're more absorbent material, they're more oxygenated, the material, and they only needed to be changed every 12 hours, where typically a tampon would need to be changed every 6 hours. These would only have to be changed every 12 hours. So incredibly popular when they came on the market. The problem was that if you bought those, and this Staph aureus can, as one of the niches that it can populate, is the genital urinary tract of women. So it's found at a reasonable frequency in the genital urinary tract. Now, just because it's sitting there, and if you happen to have it and it has this gene for the TSST1, doesn't mean you're going to get disease. And as I say, occasionally you would, and it would be very, very rare to get the toxic shock syndrome from this. But what happened was, because even if you get infection, again, it didn't mean that the gene was going to be expressed and give you disease. But what happened with these tampons is, because they were so highly oxygenated, and there was something about the material that they were made out of, that it produced an environment in which the bacteria could reproduce to very, very high levels. So if you were using them and you happen to have one of these strains, the strain would get into the tampon, it would grow in the tampon at, say, to very, very high levels. And there was something about the material and the levels of oxygen in them that was causing this particular gene to be expressed. So the gene would be expressed, so the toxin would now be in the tampon. The tampon would become soaked with the toxin, and then it would make its way into your bloodstream, and then you disease. It did not take long before they figured out exactly what was going on, and these were taken off the market almost immediately. Finally, staph food poisoning. You know, we talked about this just briefly, but let me talk about the disease itself. We don't know the mechanism. It's an enterotoxin. We know it's what we call an intoxication. An intoxication means you actually, in this case, you're going to eat the toxin. So here's a circumstance where you may have, let's say you go to a picnic. It's early in the morning. It's a hot summer day, and you have tuna sandwiches, and you bring some tuna sandwiches to the picnic. I say tuna because Staph aureus, for some reason, loves tuna. It grows very well in tuna. And so you have this tuna sandwich, you take a couple of bites out of it in the morning, you just stick it on the picnic table, you walk away and you forget about it, right? And so, you know, you've bit into the sandwich, you have some Staph aureus in your, you know, in your microbiome, in your mouth, and now there's aureus in the sandwich. And it's, you know, it's a nice warm day, so the bacteria starts to reproduce in the sandwich. And let's say that it contains the gene for, you know, this enterotoxin, so it starts to produce the toxin, and it produces the toxin in the sandwich. So now the sandwich has got the bacteria and the toxin on it. As the day goes along, it gets hotter and hotter and hotter, but it's so hot that the bacteria can't survive anymore. So the bacteria actually dies because the temperature gets actually too high for the bacteria. But the toxin is heat stable, so the toxin sticks around and it's still in the sandwich. Get to 5, 6 o'clock at night, some little kid comes wandering through. I'm hungry. So you say, okay, go eat a sandwich. The kid goes to the table, sees this half-eaten tuna sandwich, and decides to finish off the sandwich. And what they're doing now is they're eating not the bacteria, they're eating the toxin. All right, so the toxin goes straight into their gut. And within about 1 to 6 hours of eating that sandwich, that little kid is going to start experiencing severe vomiting, diarrhea, and that's going to last for about 2 days. And then the toxin will clear your system. Now, I've told you it's a super antigen, but that's not the mechanism, because the toxin never gets inside you. It just stays in your gut. And it causes the disease in your gut through some unknown mechanism. That's what we call an intoxication, where it's a chemical, you're eating the toxin directly. And the reason it's so quick, and can be diagnostic of this type of, you know, if it's a Staph aureus food poisoning, it's usually recognizable because it's so quick, right? 1 to 6 hours, usually, you know, less than, you know, usually more like 1 to 3 hours. And we've certainly had one example of this taking place in our family over the years. And as I said, it was something that was eaten in breakfast. And then we're actually in Vermont, and we're heading north to go skiing. And about 2 hours into that drive, you know, we had this terrible case of food poisoning, and it cleared up in about two days. And you don't have to treat it. There's no point in treating it because you're not infected with anything. You have no bacteria in you. You just have the toxin. So you just, you know, if it's really bad, there are antitoxins that can be used, but for the most part, hydration, you know, monitor, make sure, take some Gatorade, and it will get better. Okay, diagnosis of staph infections. If they're localized or systemic infections, you can culture the bacteria. Maybe if it's an abscess or some kind of lesion, if there's sputum, even if it's a blood sample. Staph aureus grows very easily. It's very easy to culture it. And then you're going to do a catalase test. You're going to do a coagulase test. And the last test you're probably going to do is an antibiotic sensitivity test. And that's because of methicillin-resistant Staph aureus. And I'll talk about that again in a couple of minutes. Treatment, if it's a skin infection, I told you, you know, if it's a boil, you know, that's most of it's impetigo, you're not going to give a kid antibiotics for impetigo. But if it's a boil, you're probably going to want to drain the boil and, you know, use some kind of topical treatment, put some antibiotic right into it. If the boil keeps coming back, if you get a chronic skin infection, then you might think of a systemic antibiotic to give them a full course of antibiotics. Focal systemic infections, so let's say it gets to your heart, if it gets into your blood, then, you know, again, you're going to need systemic antibiotics. Unless it's methicillin resistant, in that case, that's going to limit what you can use in terms of an antibiotic. You're probably going to use vancomycin. I'll talk about this again in the next slide or two, so don't worry about this too much for the moment. Vaccine, nothing yet, despite attempts. We thought, again, there was thought to be a really good, it would be a really great bacteria to have a vaccine against because it creates so many different diseases. But we thought we had a pretty good disease about 10 years, a good vaccine about 10 years ago, but then it turned out we did not have a good vaccine. So subsequent clinical trials, it didn't work out so good. So despite a tremendous amount of money, tremendous amount of effort, there is no vaccine for Staph aureus infection. So let me just finish this first hour by talking about methicillin resistant Staph aureus. Now, most Staph aureus, about 80 to 90% of them produce beta-lactamase, all right? So beta-lactams are really not your antibiotic of choice if you're treating Staph aureus infections, okay? Because, you know, they're producing beta-lactamase, that they're resistant. You're going to actually be resistant to a lot of these, you know, at least some of these beta-lactam type antibiotics. And so you're not going to waste your time with beta-lactams. You're probably going to use something else. That kind of resistance, however, is not methicillin resistance. Methicillin resistance is something else, okay? So methicillin resistance arose, it's associated with a gene called MEK-A. And MEK-A encodes a mutant penicillin binding protein, okay, called PBP2A. And this penicillin, remember, beta-lactams, okay, target penicillin, right? And so this is a, beta-lactams target the penicillin binding protein. So if you have a penicillin binding protein that's now mutated and no longer binds to these, all these beta-lactams, not only are you resistant to methicillin itself, you're also resistant to now a wide spectrum of beta-lactams. So methicillin resistant, this particular type of, you know, it's specific for it was initially identified as specific for methicillin, but it was subsequently shown, because the penicillin binding protein itself is mutated, that a lot of different beta-lactams don't work on it whatsoever. So this was a real problem. But an even bigger problem for this particular resistance was that it's frequently accompanied by resistance to a broad spectrum of other antibiotics. So methicillin resistant Staph aureus is not only resistant to beta-lactams, it's also resistant to other types of antibiotics. It's multi-drug resistant, and I'll show you why in a second. That's hospital-acquired MRSA. There's a new type of MRSA which was more recently discovered called community-acquired MRSA, and that's actually rarely accompanied by resistance to other antibiotics, but it's possible that this may occur eventually. So if you look at hospital-acquired MRSA, okay, this is found, where do we find this? We find it in hospitals, as the name implies. So this shows up in hospitals. Patients in hospitals show MRSA, and because of that, they cannot be treated with a lot of different antibiotics. It's generally older individuals. Generally, they have immunological deficiencies. It's generally associated with the full spectrum. It's not limited to one type of Staph aureus disease. All the different Staph aureus diseases, we see examples of methicillin resistance. In fact, as much as 20 to 35% of all hospital staph infections are actually multi-drug resistant, okay? And, you know, this is the problem here. So if you look at this particular, the HA-MRSA strain, you see that it's got this large pathogenicity island, And this island almost seems to be like a sink that attracts antibiotic resistance, right? So there's the MECA gene, but it's not by itself on this island. There's tetracycline resistance. There's streptomycin resistance. So it's got this large cassette that has multi-drug resistance. So that's a huge problem. The good news, I mean, it's large, but the good news is it's non-mobile. So it's been spreading, but relatively slowly. And it's been pretty much, I say, limited to specific strains of Staph aureus. Okay, so that's, again, a major clinical issue. Now, community-acquired methicillin resistance or community-associated is something new. First couple of cases came out about 20 years ago in 1997, 1999, but just recently we're starting to see a huge uptick in this. These are otherwise healthy individuals who've not been in the hospital for at least a year. So this is just spreading in communities. It usually associated with skin infections, okay, like impetigo, folliculitis. And when we look at these strains, okay, what we see is that we don't see this large pathogenicity of the island. We see an actual small pathogenicity of the island, and all they're carrying is MECA. So they're resistant to a lot of these beta-lactams. So beta-lactam-based antibiotics can't be used, but you can still use other antibiotics. It seems like a recombination event has removed a large section of this island and left just the MECA. So that's the good news, is you can treat this with other antibiotics. The bad news is when it lost this big piece, it became mobile. So it can now actually be passed between different strains, and it can actually potentially be passed from one species to another. So we may start to see more of this methicillin resistance, starting to spread to different bacterial strains associated with different diseases. But so that's bad because, okay, we can't use the beta-lactams, but because it's just this one resistance, hopefully it won't recruit other resistance genes to it. But for the moment, this can still be treated. There are other antibiotics that can be used. So what do you use? You use vancomycin. So that's become the antibiotic of choice. It's like if you have the hospital-acquired methicillin resistance, the only thing you're really left with is vancomycin. But again, the vancomycin story is in itself becoming worrisome. We've known for a number of years that there's something called intermediate susceptibility, which means that, individuals, you treat them with vancomycin and it doesn't work that great. It's like an intermediate effect. So now in that case, you can usually increase the amount of vancomycin and it works better. But that at least, it was kind of like a harbinger of the future. It's like, wait a minute, we're sort of halfway to vancomycin resistance here. Is it possible, we might be able to go that last little step? And sure enough, we're now starting to see full vancomycin resistance. So in 2017, there's 14 cases in the United States of full vancomycin resistant Staph aureus. That number, we're so aware of it and people are watching it so closely. We've been able to keep that number under control for the last few years. But I mean, vancomycin, that's the last line here. If we lose vancomycin as a treatment for these methicillin resistant strains, these hospitals, there's nothing left in the arsenal. And it's not like there's a lot of companies out there making antibiotics anymore. It's just not considered to be a particularly profitable business to get into anymore. So there's nothing on the pipeline. So epidemiologists and bacteriologists are watching this vancomycin resistant story very, very carefully. All right, so that's the end of my first lecture. I'm sorry, we ran about 10, 15 minutes longer than I wanted to, but I think that's because I spent more time than I was planning on spending talking about the exam. So why don't we go go ahead and take why don't we take a 10-minute break? And so it's 3:35. Why don't we start up at 3:45 with the second lecture? All right.
Dr. Whitehead, I just have a question.
Yeah.
You talked about the coagulase test and how there's the areas that's positive and epidermitis that's negative. Are we only talking about areas in this lecture?
Yes, yes. So, well, we talked about epidermidis too, right? So those are two staph strains, both caused endocarditis. So that's why I showed you how to distinguish between the two of those. So, you know, if, for example, there was an exam question, you know, that was, you know, a patient had endocarditis, and, you know, the tests were, you know, the tests may suggest that it's a staph strain. Then you would there's still two possibilities, right? And so you'd have to be able to distinguish between those two possibilities. So, you know, as I say, because epidermidis does cause endocarditis, I do hold you responsible for being able to distinguish it from other strains that cause the same disease.
Okay.
Does that make sense?
Yeah.
Yeah. Okay. So yeah, only because even though I just talked about it for that slide, I will, you know, I hold you responsible for that test and being able to distinguish. Okay.